Thank you. Well, it's really a pleasure to be here with this distinguished audience, and I'm happy to tell you some of the work that we're doing at Agenus, and particularly on a newer area for us that we think is effectively like a new dimension to approach for immunotherapy and targeting. First of all, just put up this for disclosure reasons. And so what I want to do is I'll go fairly fast through the presentation, but I want to talk about, basically, just mention what other things that we do. Yeah, looks like it needs a backup pointer. Where are we? Yeah. So the various portfolio briefly, just so you know where we are coming from. Then talk about a little bit of just a background from many people. We're very familiar with the post-translational modifications that go on in signaling, including phosphorylation, and then how we've been able to, with colleagues who really came up with this approach, take advantage of this. So these phosphoproteins are then, of course, degraded, and Tim just elegantly described, like other proteins are presented on MHC molecules, and actually these phosphopeptides can be seen by the T cells, and therefore, we're trying to exploit this both in terms of understanding the biology of how it happens and what happens in patients, and then taking that therapeutically. So obviously, what's really happening now in cancer treatment is a revolution, I think, that we're all very excited about. Conventional treatments, obviously, at least with serious, potentially terminal cancers, that would otherwise progress, have clearly shifted the time that patients have, but in many cases are not really curative. However, some of the immunotherapies, like initially CTLA-4, have had a real difference, and that is they've actually introduced, at least for the minority of patients that respond to them so far, actually very durable, and in fact effectively cures in many cases. And that's really amazing, but of course this is slightly optimistic. In many cases it's not 20-30%. In many types of cancer it's a lot less than that. But it is clear that the combinations of some of these and other immunotherapy approaches really have the potential to produce cures even in the majority of patients. So that's obviously a major change in the approach. And so Agenus' approach has been basically to pack in the portfolio with a lot of different approaches, which individually we hope will clearly have beneficial effects in some patients, but clearly the potential is for the combinations to really all work together, and if you have them all in the same portfolio, then you can much more easily do clinical trials, because you have to have companies talk to companies, and potentially you can then really get faster into the clinic with the combinations as well. And so what we have is we have a great antibody platform, which was derived from a couple of actually sources. One was a company called 4Antibody that we acquired that makes humanized antibodies on B cells by B cell presentation. So it's a FACS sorting approach, very rapid. Also, we have phage display and yeast display platforms. So we're able to make antibodies against all of the classic checkpoint modulator targets. Several of these are now in early stage clinical trials. Some in combination with collaborator Insight and other collaborators. And of course, we have a bunch of other antibodies that we're developing at this stage, at the early stages. But this is potentially very synergistic and complementary to our other approaches, which include classic approaches like heat shock protein vaccine, prophage, which is currently undergoing late stage clinical trials. And heat shock proteins are also very interesting molecules. I'll just touch on them briefly, but basically some of those are able to load, to chaperone proteins, as Tim was just talking about taposin. And some of the proteins and peptides that are chaperoned can get presented by the immune system as well. So it's a way of tapping into what is potentially immunogenic in tumors. And now we have a fully synthetic version of that using recombinant proteins, recombinant heat shock proteins and synthetic peptides. And we're combining that with the phosphopeptide approach as well. And we have some other very early stage work that we're doing in the company, but the other thing that's much more advanced is actually we have a pretty good adjuvant that's been now licensed in some GSK vaccines. So all of these approaches obviously we can combine and have potential for combinations. So this just shows kind of where these different approaches could interact. And of course, they interact at various stages, potentially. Checkpoint antibodies clearly can affect T-cell activation. You have to have T-cell trafficking to the tumor to allow it to actually kill tumor cells, obviously, and recognition. Some of these phosphopeptides I'm going to talk about seem to be, along with many other antigens, potentially good targets for the tumor recognition. Checkpoint antibodies can then act on the effector cells within the tumor, obviously. And then vaccines can be taken up by antigen-presenting cells and presented to T cells and hopefully force this cascade to work more efficiently in patients that are progressing. Obviously, in healthy people, this may be going on to a low level and effectively controls any microtumors that would otherwise progress. And I'll mention that briefly again during the talk. So this is another way of looking at what a lot of people will have seen, which is the spectrum of mutations in patients, in cancer patients' tumors, compared to their healthy tissue. And so this actually is slightly different to the very classic published version. This is looking at exomal sequencing. So here we're looking at real expressed mutations rather than just mutations in non-translated parts of the genome. But even so, you can see a very similar pattern, which is that some types of tumors have very few mutations. Now that is good in terms of using monotherapy, classical monotherapies and kinase inhibitors and such like, but it's not so good for the immune system because it means there's a lot less targets potentially to go after in the for the tumor cell to be recognized. And at the other end of the spectrum, you have melanoma and other tumors which have very high levels of tumors, like the CT26 tumor that Tim was just describing. And what we now know is that actually having a lot of mutations, and very often it's the patients who have these high levels of mutations, actually respond very well to a single monoclonal antibody against a checkpoint antibody, like CTLA-4 or PD-1. However, as you can see, that's the minority actually of melanoma patients, and it's certainly much less likely to be effective in the majority of cancer patients. So therefore, it's likely that combinations, such as combining these CPMs with vaccine approaches, is going to affect a lot more patients and be a lot more widely applicable in cancer patients, but still may not give us the handle on the majority of patients. And finally, when you combine all of these approaches, in many combinations, and particularly with vaccines helping you to target some of these epitopes more effectively, the rarer ones, it seems more likely that we're going to have that many more patients treated. So obviously tumors are very heterogeneous. Any individual patient will have a lot of different tumors with different roots, although they have common original kind of trunks, if you like, with mutations which are common to most of the tumor cells in the body. And those are the ones that obviously are most likely to be effective in actually curing a patient, rather than just dealing with an individual metastatic lesion, for example. So tumors, as we know, are dysregulated signaling machines, effectively. And so we now know a huge amount about the signaling control mechanisms of normal proliferating cells and how then these can go wrong in tumors. So we know that normal healthy proliferation circuits which have on and off switches become dysregulated, and they will continually cause proliferation of the tumor cells. And this, of course, makes them different from normal proliferating cells. So that gives us something to actually target, potentially. And, of course, some of that involves mutations in various oncogenes and tumor suppressors. But, of course, a lot of the other mutations are in completely non-driver genes of the cell. So those aren't necessarily as good targets as targets which are involved in causing the proliferation of the cell. So we reason that, therefore, that molecules and for example, phosphopeptide targets which are involved in signal transduction which was dysregulated in tumor cells would actually make tumor-specific and driver kind of targets to go for. So there's a whole series, obviously, of tumor targets that have been used over the years and still being investigated, and many of them have real potential in different situations. like oncogenes themselves with mutations, tumor overexpressed proteins, as well as the classic cancer testis differentiation type tumor antigens as well. And these obviously are, obviously giving us potential treatments and approaches, but the more targets you have, potentially the happier you're going to be. So post-translational modifications really gives, we think, a fourth dimension to target. And as I say, it's likely that a number of these will be different between healthy cells, which are normally proliferating, and dysregulated tumor cells. So they're not, first of all, going to be obviously represented in mutation by simple nucleic acids. So a nucleic acid vaccine approach wouldn't work to target these because they're not actually post-translationally modified. But they are neo-epitopes, and I'll show you that they can trigger very strong immune responses in patients. One of the other features which is really useful is that they are common, likely because some of these are involved in driving the tumors, and therefore only certain ones which are actually going to drive a tumor will actually be common between different tumor types. And of course you can't, as I just briefly mentioned, you can't really use classic approaches like live vector or RNA vaccines to try to put these things into people because they're post-translational modifications. So what we're doing in this system is basically looking for changes in tumor cells which are causing dysregulation, and including phosphopeptides and other changes. And because a small fraction of these will actually be processed and presented on MHC, these are the ones that we're interested in terms of their potential as neoantigens. So phospho-neoantigens, if you like. And of course, these will be dictated by an individual tumor cell, by the individual kinases which are dysregulated, the phosphatases control those, the stability of those modifications, the proteases, the MHC, and everything else that goes on to the whole processing pathway that we just heard about. And there are different ways that, having identified these targets, we can then potentially target them clinically. So the first version of this, and that's the version that we kind of inherited when the company acquired Phosphoimmune, was really developed by Don Hunt and his colleagues Vic Engelhard and Mark Cobbold, who was then, all three of them were then working at University of Virginia. And Don is, as many of you, anyone who knows mass spec will know Don Hunt. He's kind of like one of the godfathers and grandfathers of the whole approach. And one of the things that he developed with his colleagues was taking tumor cells, isolating by simply immunoprecipitation the MHC molecules from those tumors, eluting peptides off, as many other people have done, but then using a phosphopeptide enrichment step. Originally, when they did this, they saw low levels of these phosphopeptides in the mass spec, but of course it's very small levels compared to conventional non-phosphorylated peptides. So they'd had put in an enrichment step which allowed them to see them in much better detail and then put them on a mass spec and analyze them. So this approach, some of this data was published by them with Mark Cobbold, and this identified a remarkable series of phosphopeptides, and this is just you don't have to worry about reading any of this in examples shown here, but nonetheless, interestingly enough, there were several really nice kind of scientifically strong candidates that were identified, like phosphopeptides of MLL, which is one of the oncogenes involved in leukemic transformation, and these were actually from leukemic samples. And then the other thing you can do is you can look to see whether these phosphopeptides come from, and are they expressed in normal cells? For example, some of them were expressed in normal T cells and B cells from these particular tumors, but were also found in myeloid tumors. In some cases, myeloid and lymphocytic leukemias had some common phosphopeptides, but then some were, as you might expect, were specific to the lymphocytic or myeloid leukemias, but were not expressed in bone marrow, where obviously the myeloid cells are derived from, and again, not in normal, the majority of them were not actually in normal B and T cells. So that made them potentially promising targets. And they've now generalized this, and we're taking this further, obviously, in many different types of cancers. And so you can see that, for example, in ovarian cancer, you might find 38 phosphopeptides from the analysis that's been done so far. And then you find that some of these are actually common, also shared with melanoma and other tumors. However, there's others which are not common between the two. They're more diverse and are not shared. So it really allows you to focus on a number of fairly common phosphopeptides that are found in multiple tumors in many patients. So mentioning that MLL peptide I just mentioned, this is some of the other targets that have been popped up with these phosphopeptides, again, which sort of all make sense in terms of their known signaling functions. And MLL is particularly satisfying, as I mentioned, because it is actually found in leukemias as a breakpoint, a translocation defect. So that's one of the ways it can be activated. But it looks like it can also be activated and used by phosphorylation as a target for dysregulation of normal signaling that goes on in other tumors which don't necessarily have the MLL translocation itself. And then the other encouraging thing was that it was found that basically that T cells, even from peripheral blood, could recognize some of these phosphopeptides. These are actually healthy donors here, and actually it turns out that healthy donors are better at recognizing these phosphopeptides than patients that have been looked at. This is peripheral blood, so of course some of them could be marginating in towards tumors, but it's also possible actually that healthy people have this kind of as an immunosurveillance mechanism, and you actually have more of them in healthy patients circulating peripheral blood. And I'll talk a little bit more about that for the rest of the talk. So this actually is kind of satisfying because McFarlane Burnett a long time ago actually kind of gave some hints that this sort of approach in general for tumor immunosurveillance might be going on, and these potential driver dysregulated phosphorylation motifs that the phosphopeptides are then being presented as MHC could really represent very much the kind of immunosurveillance that he talked about. So this is a nice crystal structure that was put together with Ben Wilcox actually and colleagues with Mark and the others, and it just shows the groove here, the peptide groove, and what was found, and this is one of the example of the phosphopeptides that have been put in. This was actually crystal structure, but there's also some modelling done, of course, and they found that the phosphate group, in some cases, like this one down here, it makes a big difference whether or not it's phosphorylated in terms of the binding. So it's a little hard to read, I apologize, but there's about 158 or 160 fold difference in phosphorylation in binding affinity for the MHC molecule when the peptide is phosphorylated than when it's not phosphorylated. Sometimes the phosphate group is pointing more upwards, so it can interact with the T cell, but it doesn't have much of a contribution to the MHC binding affinity. And there are other examples where it even points down, relatively down. So that's so we're starting to model how that interaction occurs in more detail. And then we wanted to obviously to know whether or not these were specific responses to these phosphopeptides. And it was quite striking that basically that if you had unphosphorylated version of the same peptide with a lot of these T cell responses to some very interesting oncogene type molecules, you really don't see good responses at all. So you really see nice, reasonable spot forming numbers, but very clean backgrounds with the unphosphorylated peptide. This shows some more examples, actually some more of that earlier slide, but a little bit more easy to see. But the other nice thing about this data here is that it's actually they were able to stratify the patients in terms of two groups: those which had more responses to the phosphopeptides and those that had fewer, although in some cases. And actually there was a significant difference in overall survival in the two groups that they looked at, although it's obviously preliminary data. And potentially most exciting of all, it turns out you can actually see killing of real tumour cells, not just tumour cell lines with phosphopeptides added to the media, but actually real tumour cells with the right HLA recognized by phosphopeptide specific T cells and not when there's a mismatch going on. So this is now a series of a bit of data on colorectal carcinoma, one of the examples of the solid tumours that we've been looking at. And I think this is really nice because you're able to obviously look at samples with really informative material. So you can get primary tumour from the patients. And of course, there's also from the same patients, they take out a lot of colon to make sure that there's no metastatic disease or local metastatic disease in the patient. And so you can compare that normal healthy tissue, relatively healthy tissue. In fact, it's far enough away from the primary tumour that it really probably is pretty healthy. And you actually see significant phosphopeptides which are only found in the tumour. One of the other features of this work that's come out, actually is quite interesting, is that a lot of the cancer cell lines that we all know and love often have very low levels of overlap with the phosphopeptides in real primary tumour from patients. So that's a little bit of a caveat. So we're careful to focus our efforts on primary tumour material. And then the other thing is that metastatic disease is different from primary disease, and it's also different from the healthy tissue in the where the same tumour are metastasized to. And again, the tumour cell lines do not give this very good representation of what's happening in the actual tumour tissue. So we've done a series of experiments with Mark Hubbold, in collaboration with Mark Hubbold and his group, and these involve looking at the phosphopeptide-specific responses of patients comparing to viral peptides as controls. And what we found is that TILs, so actually in this case taking the tumours out, T cells out of the tumour, also can show reactivity to the phosphopeptides, even actually ex vivo. You can see a little bit of reactivity there even by fairly unsensitive ICS. It's easier, of course, by ELISpot. But when you expand them in vitro, then with phosphopeptides, then you certainly see a massive expansion of these reactive populations to a number of these different phosphopeptides. So quite good responses amongst the CD8 T cells that we'd like to see the responses to. One of the nice features of these T cells is that they are often polyfunctional, so that just like with viral peptide responses, some of these T cells responding to the phosphopeptides are actually fully capable of making all the kind of good Th1 responses we'd like to see. There's a little bit of a trend. I wouldn't make a strong point of this, but at least statistically it's there for the presence of higher numbers of these phosphopeptides in an individual tumour having stronger TIL responses from these patients. Obviously there's an outlier, so this is not large numbers as you can see. But again, rather similar to what was found in the leukemia, we can see nice killing of phosphopeptide expressing cells, which is specific to the phosphopeptide, not seen when the cells don't have a phosphopeptide or have an unphosphorylated peptide present. So it allows us to actually potentially target the tumours directly with this approach. So finally, in terms of diseases, with hepatocellular carcinoma, another obviously important disease area, and again in HCC, unlike in other liver samples, you see a significantly large number of phosphopeptides. HCC, of course, is usually typically cirrhotic. But of course there are areas which then expand as the carcinoma grows. So this is probably the closest negative control you can look at there. And again, when you look at the patients, you can actually see the responses to these phosphopeptides that you can identify from the tumour tissue. And in this case, actually, there's a trend at least towards having lower levels of responses in the HCC patients compared to in patients again, which or samples from relatively normal liver. And so this again, when you map that in terms of how it looks in terms of a Venn diagram, again, cirrhotic liver is actually within the HCC samples, so a lot less phosphopeptides. Non-cirrhotic liver has some overlap, but not complete. And then again, HepG2, one of the classic cell lines, really doesn't have great overlap with phosphopeptides found in real primary tumours. So how can this be exploited? Well, the first version of this, and I think this is a really great work from Vic Engelhard and his clinical colleagues, was to basically take these phosphopeptides, a couple of examples of these phosphopeptides that they'd identified, and give patients either one phosphopeptide a fairly simple adjuvant approach that they could use, or another phosphopeptide. And the final group after that was, those patients did okay, was actually a combination of the two. This was a small, just a phase 1, obviously it's a toxicity trial, but what was very encouraging with these minimal epitope peptides was that half the patients responded to IRS2, one of the patients that received IRS2, so it was specific to the patients who actually got phospho IRS2 that they actually responded. And likewise, some of them responded to the BCAR peptide. And then patients who'd had both, there was some overlap between the two. But only the patient any patient responded to a phosphopeptide only if they'd received that, and not the control patients, if you like. So it was a nicely controlled little study. So what we're doing now, last few minutes, basically we're taking what they've given us and what we'll continue to collaborate with them, work with them. Their laboratories are still working on this as well. So we're combining that with our own efforts as Agenus much more now, and integrating the two, and basically identifying all these different phosphopeptides that we can, and then profiling them and finding out which ones make the most sense from the scientific perspective, more likely to be driver mutations, or not mutations, but driver modifications, and which will be good to target, and then targeting them based on their ability to bind MHC and other approaches. To aid that endeavour, we have a really strong group that's involved in this work, in the mass spectrometry in particular. So Don Hunt, we've given us basically 2 of his staff, who, when they finished, the postdocs have gone on to our laboratory, which is housed just next to his university there. So they're continue to work and develop further understanding of the ligand, basically the MHC ligandome of the phosphopeptides. And then we're able to turn this into vaccines and other approaches. For example, we have the recombinant heat shock proteins as a platform, and then we have the synthetic chemistry to make the phosphopeptides and other analogues and so on in order to use those further. This is the machine that we got for Agenus South, which is basically the machine that Don Hunt would have liked to have in his own lab, so he comes over and plays with it when he can. And it is very close by. And it really allows the Agenus South group, which is, as I say, is derived from his group, to really work seamlessly with them. And so using that, it's been set up. It was set up towards halfway through last year when it got started. So now they're continuing to improve and increase the flow throughput, if you like. But I'll just show a couple of examples of what they've been doing since that and where we're taking it with that. So first of all, they can do what we call targeted ligandome. This is much quicker because this is basically, you know what you're looking for, but you want to find out if it's present in a given sample. So then you need much less sample. You can actually get away with a very limited amount of tissue, which is more of a problem for the full ligandome. Immunopurify the MHC, just to clean it up a little bit, and run the whole sample. Because you know what you're looking for, you know this peak size, so you can look at specific targets. And so when we do that approach, so far, for example, we can detect with actually as few as 10 to the 7 cells. So really not very many cells, and some of these tumor cells don't express high levels of MHC. We have, detecting most of the phosphopeptides that we would predict to find in this approach. You can also use it to look at conventional non-phosphorylated peptides, of course. And so just examples from GERCAP transduced with B7. B7 is particularly interesting for us because it's really good at binding phosphopeptides as well as other interesting peptides. But A2 also binds phosphopeptides. But these actually are just conventional peptides, preferences for binding of these two, which are coming directly out of the ligandome analysis, which agrees with published data. And as you might expect, the peptides are coming out at the right length that we'd expect. But then the real power of this approach is to take it to where you can get good quality large samples of tumour. Then you can, again, immunopurify, clean up. But as I mentioned before, our version of Don Hunt's procedure is using this IMAC phosphopeptide enrichment and then load it onto this state-of-the-art machine, and get more information, get phosphopeptides which we can then put into immunological assays. And so the results we're picking up are promising, again, because we're picking up what we'd like to see, if you like, for dysregulated signaling molecules. So kinases, phosphatases, cytoskeletal proteins, proteins that would be likely to be involved in dysregulated function, as well as in normal proliferation. And the signal transduction cell cycle dominated in terms of their assignments. So what we do is we actually have over 1,000 peptides already, and there's probably quite a lot more phosphopeptides to identify. But of course, we're only interested in the ones which are going to be tumor specific. So we rule out the ones which are found in normal tissue, of course. We want them to be present in real primary tumor, not just in cell lines. The next kind of filter we use is, are they recognized by central memory T cells of healthy individuals? And we think that's a good safety check to suggest that this is actually, first of all, safe as a target, as well as potentially a meaningful target. Actually, only about half of them have been through that test yet, so about half of those then end up going through into this experiment, and of those, a minority actually come up out of that. In leukemias, there's another set for solid tumours and some overlap between the two. And then, of course, we're focusing on certain HLAs that are common, so that brings us down to a reasonable set of HLAs that we are adding. And for example, for B7, we still have 15 phosphopeptides that we think are interesting to work on. And similar results with the other HLA alleles. And finally, how we're initially approaching this in terms of a therapeutic approach is take advantage of a platform that we've already developed, and I mentioned at the beginning, which is the heat shock protein vaccines. So the original version was derived from tumor and patient material, which was from which the heat shock proteins were extracted and then vaccinated. So this was really an individual vaccine for each patient, very different in each case. That's still in clinical trials, including now in combinations with CPMs. But we have developed the PhosphoSynVax approach. This was actually derived originally from using recombinant heat shock proteins with herpes simplex virus phosphopeptides. So that was more of a generic vaccine for anybody, and that's also in clinical trials. But for the tumor version of that, we actually take small samples of biopsies and pathology samples can be used. As long as you can get good quality next generation sequencing data out of the tumor, we can use that to get the complete phosphopeptide, but actually, in fact, any mutations as well out of the tumor, and then synthesize synthetic peptides that can be combined with the heat shock protein, recombinant heat shock protein in this vaccine format. We actually also include our adjuvant in there for good measure. So that's PhosphoSynVax, so that's the non-phosphorylated version of the heat shock protein synthetic vaccines. That's just started clinical trials, I mentioned. And finally, the PhosphoSynVax version of that is to take, basically, instead of using these mutated peptide predictions, to use phosphopeptides that are predicted from the ligandome analysis and the subsequent filtering and immunology that we do, identify a panel of phosphopeptides which we think are hopefully common to a number of different patients, maybe with multiple diseases, and then combine those again with the same recombinant heat shock protein and adjuvant. And so that will be a clinical trial that we're hoping to start early next year. And so this basically we think is a generic approach to a new dimension, if you like. For tumor targets, it basically uses all of our MHC binding information that we can. Of course, all the predictions so far that are very good for non-phosphorylated peptides, because of the data I showed you in terms of the structure, obviously are not perfect. They're not bad, but they're not perfect for phosphopeptides. And there are other post-translational modifications as well, which are interesting, that also come out of this mass spec analysis. So we do think that by using mass spec, we can really get at potentially important phosphoantigens. And by showing that patients and even healthy people can actually respond to these, we have the potential for improving that response in the patients. So there's different ways of, and I showed you the first example, we have the PhosphoSynVax approach to actually taking that into the clinic. So I really want to thank all the people who are doing this, and this is the founders, PhosphoImmune founders, Mark, Don and Vic, who are great colleagues. We meet with them all the time. We're going to visit them next month. And Mark actually came back to the UK for a little while, like myself, but then he got headhunted back to Mass General, where he is now. And the other guys are still at the University of Virginia. And then, of course, we have our own internal team, some of whom I mentioned as I went along. Thanks very much.